Hello, I am Eli Adashi, Professor of Medical Science at Brown University and host of Medscape One-on-One. Joining me today is Dr. Harold Varmus, Director of the National Cancer Institute of the National Institutes of Health, a 1989 awardee of the Nobel Prize in Physiology or Medicine, a former director of the NIH, and until recently, the president of the Memorial Sloan-Kettering Cancer Center. Dr. Varmus is hardly in need of introduction. Welcome.
Thank you.
It's wonderful to have you and to meet you after all these years of observing you from afar. December the 23rd will mark the 40th anniversary of the National Cancer Act and the initiation of the war on cancer, if you will. To the extent that you can you give us an overall sense of what we may have accomplished over the last 4 decades?
Well, first of all, I think we've changed the metaphor. It's inaccurate, in my view, to think about a war on cancer as though cancer was some single individual enemy. Nor is the metaphor of war exactly right. So we now understand that cancer is actually a constellation of diseases, many different diseases arising in different tissues. Indeed, the number of diseases that cancer represents is only multiplied over the last 40 years as we understand more and more about how cancers arise. Secondly, we understand that cancer is an outgrowth of some fundamental principles of biology, how genes control our development, how development goes awry, how different genes can influence the initiation and progression of cancer. So the consequence is that we have a much deeper understanding of cancer as, in a sense, a byproduct of the way life is organized. And we have a sense that there are many puzzles to be solved. When we think about progress, which is the question I'm always asked, yes, we've made enormous progress against some diseases. We've made much less progress against others. And the challenge for us is to define the problem more clearly so that we can begin to make more rapid progress against some of the diseases against which we have prospered very little.
Upon taking the helm, you outlined 5 priorities for improving the cancer research program. Amongst those, you proposed to reform the clinical trial system. Mm-hmm. What concerns were you hoping to address, and how is this effort going?
Well, there were 2 major issues. The first had to do with what frequently happens to a complicated system that's been in existence for a very long time. Namely, its organization is no longer well suited to the kinds of trials we're trying to run. So with the help largely of one of my chief deputies, Jim Doroshow, the clinical trial system has been dramatically reorganized. The number of groups will be reduced from roughly 10 to 5. Many of the groups that had a rather singular focus are now embedded in much more complex organizations. And the idea is to have virtually every one of these groups, a pediatric group and 4 adult clinical trials groups, taking on a wide variety of conditions, because we believe that there's a lot to be learned from observing what's going on with other cancers when you're focusing on your own. The second big issue was the organizational issues were also directed towards speeding up the process of getting trials going and carrying out trials and deciding when trials shouldn't be finished and making sure that we're not losing the proper speed in responding to new advances in science. But the second big issue is one of incorporating more scientific thinking into the way trials are conducted, so that trials are not simply tests of whether a drug works, but they're tests of biological principles. And when a trial does not work, we learn something about why it's failed that's actually instructive about the next trial. So we think that can happen, and we've gotten the leaders of the clinical trials groups more closely aligned with clinical center directors and other investigators in the scientific community to be sure we're collecting the right samples and carrying out the trials in a way that is consistent with new developments in oncological science.
In a sense, this was the other priority you outlined, developing new questions, if you will, and new scientific agendas that you wanted to pursue at the time.
Well, but that's not the way we're putting that into place. So the latter part of this is very important to me, and that is to get the scientific community engaged. And by the scientific community, I mean everyone, not just those people who work at laboratory benches, but clinical scientists, behavioral scientists, other folks who are working on the cancer problem from an epidemiological point of view, from a medical care viewpoint, and to begin to define what aspects of cancer science are still deficient in knowledge. We have called the questions that come out of that kind of exercise provocative questions, and over the last year we have compiled an interactive website that people can go to by just typing in provocative questions on your Google searcher. And you will see that we've now advertised for applications to try to answer what we consider to be, so far, the 24 best questions. We've received close to 1,000 letters of intent to apply to answer these questions, and we think this is a very novel way to use our resources in an effective fashion at a time when, quite frankly, budgets are not only not increasing as they used to, they're decreasing.
Much has been made of the hoped-for impact of the Genome Project on the both diagnosis and treatment of cancer. Are we reaping some benefits?
Oh, tremendous benefits.
Are we hoping for more?
Well, that always goes without saying, but the fact is that people at first probably oversold how quickly genomics was going to lead to changes in medicine, and then there was a reaction, which I think is very unfortunate, of people having the kind of dubious tone you had in your voice, because in fact the Human Genome Project has, first of all, dramatically accelerated cancer research on many fronts. Secondly, it actually has already transformed the way we think about cancer. When I said earlier that there are many more categories of cancer than we knew before, it's because using the tools of modern genomics and just DNA sequencing and cloning, we have subdivided cancers by the nature of the lesion. It's a little bit like saying at one time we knew there was pneumonia, and then we were able to identify specific bacteria and say there was E. coli-induced pneumonia, and pneumococcal pneumonia, and E. coli and tuberculous pneumonia. The same thing has happened in the impact of genetics on cancer. So now we know that whereas once there were 4 histological types of lung cancer, now we can say that for adenocarcinoma of the lung, there is a type that is dependent on EGF receptor mutations, a type that's dependent upon ALK gene fusions, there's a type that's dependent on Kirsten-Ras mutations, and on, because there are actually quite a few categories, some of which actually are prognostic indicators, and more importantly, some of them are now treatable, not totally successfully, but with at least partial success, with drugs that inhibit the specific proteins that are mutated as a result of the mutations I've mentioned.
So personalizing the treatment to the extent
making it more precise.
Yes.
I think that's the better term. Personalized to me is a kind of euphemism. It's not very specific. My father practiced personalized medicine, but we're trying to be more precise, and I think that's an incredibly important issue, and it's a way in which genomics in particular, but also other kinds of science, are transforming diagnostic categories. And over the next 10 years, not only in cancer, but in many fields of research, as a report from the National Academy of Sciences recently indicated, all of our nosological classifications are going to change. But in cancer, it's already happening. And those changes affect what we call a cancer, what we think will happen to the patient who has it, and how we're going to treat the cancer. And it may even begin to affect how we prevent it. Certainly, it's going to influence how we screen for it.
From where you sit, how well are we doing as a nation in bringing innovative therapeutics to the cancer table?
Well, it's slower than one would like, given the fact that we now have so many potential targets identified. But as the FDA recently pointed out, they've approved more new drugs this year than ever before, at least before in recent memory. Most of those drugs are cancer drugs. And in my own field of cancer research and therapy, there are some tremendously positive hits. And we're clearly far from being able to treat in a curative fashion as many cancers as I'd like to think we can cure, but there are many for which we now have important drugs with important anti-cancer properties. It's also important to point out that we have much better ways to control some of the side effects of cancer, better control of nausea, of pain, of bone marrow toxicity. Sometimes this gets forgotten in our lust to control the cancer, but making cancer a disease that you can live with and go to work with, increasingly, as I learned, especially during my time at Memorial Sloan-Kettering, we have many patients with lethal cancers who are actually feeling pretty good and are working full time and enjoying their families. And as long as their symptoms can be kept under control by radiotherapy and drugs that control symptoms and other modalities, we're doing right by our patients.
Critical. One way to measure our progress or success is by monitoring the national mortality cancer rate. What's the latest in terms of the trends that we are monitoring or surveilling nationally?
Well, the overall trend is good. We have been seeing roughly a 1% decline in age-adjusted mortality overall for all cancers, men and women, over the last 10 years or so. And some important signposts have been passed. For example, we expected to see decline in lung cancer mortality for women because they're now starting to stop smoking, whereas 30 or 40 years ago they started to start smoking. And just this last couple of years, we've seen the beginnings of decline in lung cancer mortality for women. Now, one of the things that we learn from such registration data is that some cancers are declining with respect to mortality rates faster than others. Some are increasing, and we have to pay special attention to those where our efforts to control the cancer are not as fruitful as they have been for others.
If you had to pick one recent, highly promising scientific or clinical breakthrough in the cancer arena.
But I don't have to do that.
You don't have to. But did something strike you as particularly exciting, or would you rather not comment on that?
Give me at least three. So in the prevention arena, or perhaps screening is a better term, we finally have evidence that helical CT scanning, low-dose helical CT scanning, can prevent death from lung cancer. Not simply detect a lung cancer and allow someone to live a long time. That's not proof that it actually affects mortality rates. But we have evidence from a very large-scale study that was ended just about a year ago because we had reached the point of showing over about 8 years that by screening heavy smokers at an elderly age, between ages of 55 and 74, that we could reduce lung cancer mortality rates by about 20%. Now that creates other kinds of problems we need to deal with. How do we actually implement the screening process in a way that's effective and affordable and acceptable to the community, and maintain the high rates of reduction in lung cancer mortality that we've seen in a very highly controlled study. But it's very encouraging because lung cancer is the biggest killer. Second thing is we've had some of the most impressive demonstrations of the effectiveness of immunotherapy that we've ever had. So we've, of course, had antibodies for some time, and that's a kind of immunotherapy, and antibodies like Herceptin clearly have benefit. But seeing that an inhibitor of the activity of the immune system, a drug that's now called ipilimumab in metastatic melanoma, is a very significant breakthrough, and it makes us feel that we're on the right track and making substantial investments in the immunological response to cancer. And then, I suppose a third example is the depth of knowledge that we're obtaining by systematically analyzing literally hundreds of tumors of each known histological type for the genetic damage that characterizes those types of cancer. And through a very large project called the Cancer Genome Atlas that we're carrying out with the National Human Genome Research Institute, we have accumulated an incredible catalog of mutations that is, in a sense, the substrate, the material that we're going to be using to learn how to prevent and treat cancer more effectively in the future.
Impressive. As we speak, about 600,000 or so Americans die every year of cancer, which would be second, I suppose, only to heart disease at this time.
Mm-hmm.
Going back to the anniversary we were marking earlier on, and I know it's difficult to project, but if you had to guess what figures...
Again, I don't have to guess.
You don't have to, but to the extent that you care to, what figures might we be looking at on December the 23rd, 2021?
Well, I'm not going to make that kind of projection. I do think there's every reason to believe that mortality rates will continue to decline.
Mm-hmm.
You have to remember that everybody dies of something. And as we eliminate other diseases, obviously others may, because cancers are in general age-related. So counting the total number of cancers is not the best way to analyze this problem. The way to analyze it, just as we do for heart disease, is to look at age-adjusted rates. And when we make that kind of calculation for heart disease, we can see that over the last 50 years, there's been roughly a 70% decline in age-adjusted mortality for heart disease and cardiovascular disease and stroke. That is remarkable, but of course it's still the major killer. So we have to factor that in. But I do think that we will be improving the age-adjusted mortality rates for most cancers over the next 10 years. Will we have major breakthroughs? Well, they're breakthroughs because we can't predict them. That's why we call them breakthroughs. So I don't make that kind of prediction. But we have an incredible amount of information. We have strong support. I wish the support were growing, but we do have $5 billion a year to spend at the Cancer Institute, and I plan to spend that well. And I am optimistic because of the strength of the science that we do that we will make significant progress. But I'm reluctant to say what progress will occur or what the number is.
Right. Fair enough. On a personal note, what is it that drew you to this challenge at this time in your life?
You think I'm too old to do this kind of work?
Well, your wife may have argued that it may be time to take it easy, but maybe she didn't.
I'm not somebody who takes it easy. She knows that, and if I try to take it easy, that's not healthy. No, I had been at Sloan-Kettering for 10 wonderful years, and we did a lot of great things there. Mm-hmm. But these jobs have a lifetime. When you're at an institution and you have ideas for it, you usually, if you're going to get them done, you usually do that within the first 5 to 8 years. And I think I did that at Sloan-Kettering. And times became tight economically, and I could either have retreated to my own lab and continued to work in my lab, which is something I enjoy doing, or I could take on another leadership position. And frankly, as I've said many times, when I was here at the NIH as the NIH director, I was frequently frustrated. The NIH director has very little money, has very little authority over scientific programs, always pleading with the institute directors to follow his or her example. And running an institute always looked like more fun. And indeed, it is more fun. I'm running programs. I'm making decisions that affect science directly rather than making political judgments and simply interacting with what's now an increasingly refractory Congress. So I love this job, and I'm very glad I did it.
Well said. Thank you. On this note, sincere thanks to Dr. Varmus and to you, our viewers, for joining Medscape One-on-One. Until next time, I am Eli Adashi.